Hi! Today, we'll take a quick look at how do you confirm you cloned a target gene onto bacterial cells using transformation in order to obtain a protein of interest. In this case, we used a PGX GFP construct onto E. coli competent cells. First, we pour already presumed to be transformed cells into an LV agar plate containing a proper concentration of the selective antibiotic, in our case, 50 micrograms per milliliter of ampicillin. We let them grow for 1 day, and if we're lucky, we got ourselves some colonies. For confirmation, we take one of these and culture them into an LV broth with the same antibiotic. Leave it 1 night, and then we extract our plasmid using whether a Telt or an alkaline particle. Here, we'll explain the first. We pellet a 1.5 milliliter aliquot of an overnight LV cultured at 30,000 RPM for 2 minutes and resuspended thoroughly in 100 microliters of resuspension bottle, which contains 15 millimolar tris hydrochloric acid pH 8, 10 millimolar EDTA, and 100 micrograms per milliliter RNase A. We then add 200 microliters of alkaline lysis solution, which contains 0.2 normal sodium hydroxide and 1% SDS. We invert gently to lyse the cells and incubate the mixture for 3 minutes at room temperature. After lysis, we add 150 microliters of chloroform, vortex briefly, and centrifuge the sample at 13,000 RPM for 10 minutes to separate the phases. We carefully transfer the clear upper aqueous phase to a fresh tube. Mix it with 2.5 volumes of ice-cold absolute ethanol and centrifuge it at 30,000 RPM for 10 minutes to precipitate the plasmid DNA. We wash the resulting pellet with 500 microliters of 70% ethanol, centrifuge again at 30,000 RPM for 5 minutes, air dry it for 10, and finally resuspend it in 40 microliters of nucleus-free water for downstream analysis. Finally, we digest the product with the very same enzymes we've been working with. Again, overnight. And now for the moment of truth. We prepare an electrophoresis gel for our pure vector, with no insert. Our pure insert, of course, our construct, both digested and undigested, and a molecular weight marker. If we did everything right, which might not be our first attempt, we will observe 2 consistent bands in our digested construct well. And congratulations! You are now the very proud owner of transformed bacterial cells. This has been a tutorial by the guys here at the Biotechnology Engineering School, Catholic University of St. Mary, Peru. Thanks for watching.